Hello everyone, welcome to ITLS Academy. Our topic of today's discussion is the embryonic stem cell. The embryonic stem cell. First point about the embryonic stem cell is an embryonic stem cell, in short ESL, is defined by its origin. The origin of any stem cell will define the properties that what kind of this particular cell is going to be. Next point is it is derived from the blastocyst stage of the embryo. As the name indicates, embryonic stem cell, it is clear that it is somewhat related to embryo. So, it is derived from the blastocyst stage of the embryo. Blastocyst is a stage at which stage implantation in the uterus occurs. The blastocyst, we can say that it is a kind of stage of embryonic development, and it is a stage just prior to implantation in the uterine wall, or we can say this is an event of implantation also. Next point, embryonic like stem cell. Remember, this is embryonic like, not embryonic. So embryonic like stem cells, called embryonic germ cells, can also be derived from primordial germ cells. These primordial germ cells are the shells of the developing fetus from which eggs and sperms are formed. So we can say that primordial germ cells are the type of cells that give rise to gametes of the body. And we know that gametes are eggs and sperms. Sperm in male and eggs in female are our germ cells, are our gametes, and these gametes are formed by, or these gametes are derived by primordial germ cell, and these primordial germ cells are also a kind of embryonic stem cell, or we can say that these are embryonic like stem cells. Here, these primordial germ cells are discussed about the mouse and human fetus. Remember, in the case of mouse and in the case of human fetus, these primordial germ cells will always give rise to eggs and sperms, or we can say to the gametes. So in this particular lecture, our discussion will be limited to mouse embryonic stem cells. We will not discuss about human embryonic stem cell. We will focus on mouse embryonic stem cells in this particular lecture. And also we will see complete details of the mouse embryonic stem cells. Here comes embryonic stem cells. You can say this stage is blastocyst. This stage is known as blastocyst. Blastocyst is a stage If we talk about the blastocyst, this is a covering of blastocyst known as trophoblast. This trophoblast is outer covering. This blastocyst also contains some kind of cells located only on the one side of blastocyst. These cells are known as inner cell mass. These inner cell mass are responsible for giving rise to embryonic germ-line cell. As we know that germ-line cells give rise to our germ-line like ectoderm, endoderm, mesoderm, etc. So now we are going to see detail of this blastocyst. Embryonic day, you can see here mouse and human. This particular cell give rise to, this is embryonic stem cell. This is pluripotent cell. As we have discussed in our previous lecture, totipotency, pluripotency, multipotency and also unipotency. If you have not watched, I will refer you to that particular lecture so that you can easily understand the things, because that lecture is a basic of all the systems we are going to study in our this particular lecture series. In short, we can say that pluripotent cells are a kind of cells that can give rise to all cells in the body. But these pluripotent stem cells cannot give rise to trophoblast. This is inner cell mass. This inner cell mass can give rise to ectoderm, that is outer covering, mesoderm, that is middle layer, and endoderm is internal or internal covering of our body or of the mouse body. Let us see a few points regarding this particular trophoblast layer. This trophoblast layer is also known as trophoectoderm, as it is outer layer, that's why it is trophoectoderm. This trophoectoderm is extra embryonic tissue placenta and umbilical cord. This forms extra embryonic tissues, and extra embryonic tissues are placenta and umbilical cord. So now we will see location of embryonic stem cells, where these embryonic stem cells are located. So the very first point is, some scientists argue that embryonic stem cells do not occur in the embryo as such. What this point exactly means, let's see. In this particular line, focus is on the point that embryonic stem cells closely resemble the cells of pre-implantation embryo, but are not in the fact the same. These are approximately same as the pre-implantation embryo, but in fact these are not same. This is a topic of argument for some scientists. If we talk about our next point, These cells proliferate extensively in the embryo. As these are embryonic stem cells, so of course they will proliferate in the embryo and are capable of differentiating into all the types of cells that occur in the adult and can be isolated and grown ex vivo or we can say it outside the organism where they continue to replicate and show the potential to differentiate. The embryonic stem cell derived cells can potentially be used to replace or restore tissues that have been damaged by disease or injury, such as diabetes, heart attack, Parkinson's disease, or spinal cord injury. So let's see some defining properties of an embryonic stem cell. What are the characteristic features? What are the properties by which we can easily understand, we can easily study the embryonic stem cells? So let's see some properties, that is defining properties. The very first point is derived from the inner cell mass. If we talk about the blastocyst, we have seen that this is the trophoblast layer and these some kind of cells are present in this particular covering. These are known as inner cell mass. So these embryonic stem cells are derived from the inner cell mass. This inner cell mass is also known as epiblast of the blastocyst. This is structure of blastocyst. If we talk about the next point, so these are capable of undergoing an unlimited number of symmetrical divisions. These embryonic stem cells undergo symmetrical division as we have seen in the formation of in the division of zygote. The zygotic cells undergo symmetrical division. So these embryonic stem cells are also capable of undergoing and symmetrical divisions. And these divisions are not limited, these are unlimited number of divisions. And here a main property of this division is without differentiating. There will be no differentiation. That point is exhibit and maintain a stable or full or we can say diploid normal complement of chromosomes. There will be no any loss in chromosome numbers. For example, if we take an example of humans, so there are number of chromosomes are 23 sets. So the number after these unlimited divisions, still it will be 23 sets of chromosome. There will not be any kind of alteration. Next point, Pluripotent embryonic stem cells can give rise to differentiated cell types. These differentiated cell types may form digestive system, circulatory system, nervous system, and many more differentiated cell types. Next point is these are capable of integrating into all fetal tissues during development. These can integrate in all fetal tissues. Next point is can be induced to continue proliferating or to differentiate. These embryonic stem cells have the property to continue proliferate or they can also differentiate continuously. Next point is lacks the G1 checkpoint in the cell cycle. This is most crucial step in their unlimited number of divisions because they lack G1 checkpoint. So it becomes very easy to proceed in the cell cycle. Embryonic stem cells spend most of their time in the S phase of the cell cycle. This is also a very important point that most of the time they remain in S phase, and during this S phase they synthesize DNA. As we know that S phase is the phase of cell cycle in which replication of DNA takes place. So here some characteristics of embryonic stem cells are shown in this particular diagram. You can see this is blastocyst. This divides symmetrically forms stem cells. These stem cells are pluripotent in nature and further they give rise to many kind of cells, many kind of systems. Pluripotent stem cells have the ability to form endodermal organs, mesodermal organs and also endodermal organs. This is ectoderm, mesoderm and endoderm. Let's see all these processes in detail. The very first characteristic is origin. These are derived from pre-implantation or peri-implantation embryo. If we talk about their property of self-renewal, so the cells can divide to make copies of themselves for a prolonged period of time without differentiating. Next characteristic is pluripotency, and we can say that this pluripotency is the most important or most crucial characteristic of embryonic stem cells. Embryonic stem cells can give rise to cells from all three embryonic germ layers, even after being grown in culture for a long time. You can see here example, this is ectoderm, mesoderm and endoderm, all three germ layers. Ectoderm give rise to brain, spinal cord, nerve cells, hair, skin, teeth, sensory cells of eyes, ears, nose and mouth, and pigment cells. If we talk about the mesoderm, so mesoderm give rise to muscles, blood vessels, connective tissues and the heart. Here comes ectoderm, this is endoderm. Ectoderm we have already discussed. So endoderm gives rise to the gut, pancreas, stomach, liver, lungs, bladder and germ cells, that is eggs or sperms. A question arises here that are embryonic stem cells truly pluripotent? So the answer is pluripotency. That is the ability to give rise to differentiated cell types that are derived from all three primary germ layers of the embryo. These three germ layers are endoderm, mesoderm and ectoderm, is what makes embryonic stem cells unique. So, we can say that these embryonic stem cells are truly pluripotent because they can give rise to all three germ layers. How do we know that these cells are indeed pluripotent? Is there any answer? So, let's see in our next point. Laboratory-based criteria for testing the pluripotent nature of embryonic stem cells derived from mice induced three kinds of experiments. So, we are going to see this pluripotency in different three kinds of experiments. So, let's start. One test is conducted by injecting embryonic stem cells derived from the inner cell mass of one blastocyst into the cavity of another blastocyst. This was an experiment in which, if we see, this is blastocyst and this is inner cell mass. So the embryonic stem cells derived from this particular inner cell mass of one blastocyst injected into the cavity, this is the cavity, injected into the cavity of this particular blastocyst. So let's see what was the result. The combination embryos are then transferred to the uterus of a pseudo-pregnant female mouse, and the progeny that result are chimeras. What this particular transfer resulted in? So, these chimeras, if we talk about this chimera are a mixture of tissues and organs of cells derived from both donor embryonic stem cell and the recipient blastocyst. This test has been extended in studies designed to test whether cultured embryonic stem cells can be used to replace the inner cell mass of a mouse blastocyst and produce a normal embryo. If we talk about the second test, so a second method for determining the pluripotency of mouse embryonic stem cells is to inject the cells into adult mice, and this was under the skin or the kidney capsule that are either genetically identical or are immune deficient, so the tissue will not be rejected. When examined under a microscope, it was noted that these tumors contain cell types derived from all three primary germ layers of the embryo, that is endoderm, mesoderm and ectoderm. Thus, embryonic stem cells that have been maintained for a long period in vitro can behave as pluripotent cells in vivo. A third technique for demonstrating pluripotency is to allow mouse embryonic stem cells in vitro to differentiate spontaneously or to direct their differentiation along specific pathways. Within a few days after changing the culture conditions, embryonic stem cells aggregate and may form embryoid bodies. In many ways, embryoid bodies in culture dish resemble teratoms that are observed in the animal. Embryoid bodies consist of a disorganized array of differentiated or partially differentiated cell types that are derived from the three primary germ layers of the embryo, the ectoderm, endoderm and mesoderm. So all the three techniques, all the three tests resulted in the formation of all the three germ layers. So we can say that these embryonic stem cells are truly pluripotent. The techniques for culturing mouse embryonic stem cells from the inner cell mass of the pre-implantation blastocyst were first reported 20 years, and versions of these standard procedures are used today in laboratories throughout the world. So thank you guys. This was all about the embryonic stem cell. The remaining things we will discuss in our next video. Thank you for watching. If you like this video, then guys please like it, share with your friends and also subscribe our channel ITLS Academy. You can also follow ITLS Academy on various platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, etc. So thank you. Subscribe ITLS Academy.